This film is an example of connection between the scientific world and the rural community through the implementation of a participatory action research in Madagascar. Testing vermicomposting on a firm scale was the subject of participatory researches carried out over several years in one area located on the highlands. The goal was to know whether vermicomposting could be a new opportunity to diversify farmers' usual organic fertilization. Cattle manure, the current local main fertilizer for crops, has an average low nutrient levels, and many farmers don't have cattle to produce it. The municipality to which we refer is Vermicompost, a dynamic area with a high agricultural potential, located only 40 kilometers from the capital city under high-altitude tropical climate. Vermicompost stands out for its urban expansion and economic dynamism. Agricultural activities are highly developed, with a strong market orientation from the cultivation of irrigated rice through diverse vegetables and livestock raising to particularly cattle, small ruminants, and poultry. These activities provide rich and locally available resources that a farmer recycles intensively, both at farm and municipality levels. Unlike many other situations in small-scale family farming, this valorization of local resources is part of a circular economy of available organic biomass targeted to crop fertilization. For example, the byproduct of rice, the main crop in the region, are the subject of financial transactions. These financial transactions involve bread producers who use rice husks as a combustible. The residual ash is then used by farmers to fertilize their crops.
The proximity of this municipality to researchers located in the capital has enabled the implementation of an original approach called participatory action research. This is essentially guided by the contextual needs leading to changes in agricultural practices by engaging the farmers from the beginning into the innovation process based on local and scientific knowledge and in learning by doing. In this participatory action research program, 3 types of activities involving the vermicomposting innovation process were carried out. First, a diagnosis at municipality and farm levels was conducted to assess the availability of plant organic resources, local earthworms, and usual fertilization practices. A network of farmers was always involved in all these phases. Technicians from the NGO called Amades permitted to train these farmers in vermicomposting. This diagnosis also allowed the establishment of field experiments and greenhouse studies. Next, evaluations of all the experiments were assessed on the basis of field measurements and group workshops.
After the diagnosis phase and in preparation for the experiments, a dedicated training related to vermicomposting was provided to the farmers on the farm school on the NGO Amades, which is involved in the participatory action research process. The farmers selected were able to visit the NGO's farm in Madagascar and benefit from its experience. To produce vermicompost, various biomass such as banana leaves and the trunks, rice straw, and eggshells are first crushed to create a pre-compost. These biomasses are before pre-composted for around 15 days. Then, they are placed in the vermicomposting unit. The earthworms feed on these substrates and transform these materials into vermicompost.
The vermicomposting process also takes about 15 days.
The important thing to consider is the moisture control.
We always to keep it being too dry.
Before being used in the fields, the vermicompost is carefully sieved.
All these steps in the production of vermicompost mentioned earlier have been shared with the 42 producers. The training provided here allowed them to learn and especially to set up their own vermicomposting units in their respective places. Then the vermicomposts produced by these farmers were tested in the field.
The farmers were then trained in how to make vermicompost. A multi-local experiment involving 11 plots spread across 6 villages in the commune was set up. Manuel Minwarsan describes the different phases of the experiment.
In the first phase, we organized a workshop to ask farmers how they to use vermicompost, if it was available to them. The farmers were free to choose the treatments, amounts and methods of applying the vermicompost. The objectives of the multi-local experiment were to compare usual practices and innovative practices using vermicompost on rain-fed rice. At the end of the workshop, a list of treatments to be compared. That is the usual practices on the one hand and innovative practices on the other, was proposed by the farmers and will be installed on the 11 experimental plots. According to the list, farmers are used to making combinations, whether in usual practice or innovative practices using vermicompost. In the second phase, the trials were set up in the field. During the trials, the organic fertilizers were applied by microdosing in rice hulls at the time of sowing. The variety used was the reference variety most commonly used in the region, which is the Chomrongdan. To give you an example, on plot 1, the farmers suggested comparing rice husk ash with cattle manure, which is the usual practice, against rice husk ash with vermicompost, which is the innovative practice using vermicompost.
The evaluations were carried out on 10 plots because an incident occurred on 1 plot. The assessments were carried out individually in each farmer's field. The assessments were based on visual observation and the farmer's perception. The individual assessments were carried out in 2 phases, one towards the end of the vegetative phase and one during the harvest. The most significant result during the individual evaluations was that the practice integrating the innovation showed more advantages compared to the usual practices, meaning that the plants on the plots receiving vermicompost had more tillers, greener leaf color, and higher numbers of fertile tills. The plants were also at more advanced stage, with a faster emergence of the spikelets, longer panicles, and less empty greens. Farmers also mentioned that plants were more resistant to drought on plots that had received vermicompost. Collective evaluations were then organized during specific workshops, where the results of all the trials carried out on each farmer's plot and those of the research were shared. In considering rice yields, 8 out of 10 plots showed that the yield with practice using vermicompost was higher compared to the yield with usual practices. The yield increase ranged from 0.24 to 1.45 tons per hectare. These differences were significant on 3 plots. These results, therefore, show that the assessment based on the farmers' visual observations are consistent with the evaluation from scientific measurements. Now, we shift into the lab environment of scientific studies, with researchers working on vermicomposts and the earthworms. They share their latest outputs and overall experiences and their contribution for a sustainable development.
The earthworms used to produce vermicompost are classified as epigeous. These earthworms typically feed on fresh organic matter on the soil surface. Here in Madagascar, two types of earthworm are used to produce vermicompost: imported earthworms and local earthworms. Two species of earthworms are the most well-known, Eisenia andrae and Eisenia foetida. These species are more active in fresh organic transformation and reproduce more quickly, so they produce vermicompost more rapidly. However, these imported species are expensive on the market, and many farmers cannot afford them. That is why we propose the use of local worms as an alternative. Local worms can be used to replace imported worms, but are less prolific and take a long time to produce vermicompost. However, the quality of the vermicompost is quite similar. Their recognition can be based on very simple criteria that farmers can easily identify. We've produced a book to help farmers recognize them. In these books, there are very simple characteristics that really permit to recognize them.
Greenhouse studies were carried out by Tukiran Jiansu as part of his master's degree and currently for his thesis. Those studies showed how vermicompost can improve resistance to water stress.
The objective of my study was to determine how vermicompost and local worms could contribute to improving the climatic resilience of rain-fed rice systems. In Madagascar, as in many other countries, climate change is increasing the risk of crop drought periods. This is why it is crucial to develop and propose practices to farmers that would enable them to better cope with these drought periods. So, as you see here, my work consists of carrying out greenhouse studies to evaluate the performance of vermicompost and earthworms in improving the ability of rice to cope with periods of drought. Preliminary results suggest that, compared with the manure traditionally used by farmers, vermicompost has allowed for better rice growth, both in stressful and normal conditions. Moreover, this effect on the plant can also be explained by the presence of substances similar to soil's phytohormones, which are secreted by earthworms and are also present in vermicompost.
A field study was also carried out by Unzaratsatusk to test several fertilization practices.
A study was conducted to test various fertilization practices, including vermicompost and cattle manure, which is usually used by farmers to fertilize their crops. To do this, we assessed from sowing to flowering the agronomic parameters, such as rice yields and cycles, attacks by crop insects in the experimentation plots. The results showed that with vermicompost, we had better agronomic performance, that is a higher rice grain yield, but also a higher straw yield. In addition, with vermicompost, we have also seen that the rice cycle is shortened from seedling to flowering. This can be up 10 days. This parameter is really very important because, in the context of climate change, where there has been a delay in the arrival of the first rain, or sometimes disruptions in the amount of rain, the rice production can be secure, meaning it can also provide higher yields. As far as bioaggressors' evaluation for disease or insect attacks, we found interesting results. I'm going to show you photos showing the comparison between different types of plot. Here, we have several plots placed side by side. On one side, a plot fertilized with a mixture of cattle manure, compost, vermicompost and poultry droppings, a mixture with pig manure and ash. Next to this is the vermicompost alone. And on the far right, we have the cattle manure with dolomite powder. These plots were subject to bioaggressors attacks, except for the vermicompost, where the biomass is really very green.
A previous diagnosis on available plant resources for vermicomposting during interviews and collective workshops identified 46 plant species that could be used for biomass valorization for vermicomposting. The farmers' knowledge on the 6 plant resources were presented now.
Buddleia madagascariensis repels insects. It takes time to decompose, so it's an effective plant source for earthworms.
We use Vernonia appendiculata.
It's easily degradable. Its abundance of liquid matter plays an effective role in easily decomposing the other plant components.
They are less available in winter, in June and July.
Straw could be used with earthworms.
The reason we use straw is that it's available during the harvest season.
Straw is easily decomposable due to the self-generated heat during pre-composting.
It's very effective at killing pests, such as white grubs. Apart from straw, we can also use specific grasses with soft stems.
Various specifically soft grasses are the most commonly used. These species are available all year round. Banana trunks are available all year round.
It retains moisture.
Rich in water, it helps in the rapid decomposition of manure.
Its advantage lies in its bitter taste, which repels insects.
The bitter taste of Tithonia never goes away, even when transformed into a fertilizer. Tithonia-based compost contains no living animal species.
It is only available from December to May. On my farm, I have local worms such as Cancangela and Cancamena. And I also have imported worms, Icenia.
The Icenia is more robust. Its color is tiger-striped red and gray.
I can recognize them by their color. It's red with a flat belly.
The Cancangela is very dynamic, whereas the Cancamena and the Icenia are less active.
The Icenia is gentle, but when you touch it, it moves a lot.
We can always get vermicompost throughout the year. If we compare cattle manure and earthworm manure, the vermicompost is of better quality because their food is very diversified, including various green leaves associated with cattle manure. So, to have significant results in research action, we have seen that the involvement of different actors is required: producers with their local knowledge, NGOs with their field experience, and the researchers with their advanced scientific knowledge. And to conduct research that aims to set up an innovation process, the involvement of various scientific disciplines, hence multidisciplinarity, is really essential. In the case of the research based on the vermicomposting innovation, for example, the intervention of botanists, agronomists, ecologists, and also experts in the human sciences was necessary in order to combine knowledges and know-how on plant resources, earthworms, vermicomposting for a better management and valorization.
Research action involves a participatory approach to produce an innovation, i.e., an innovative solution. It proceeds through different phases. As we have seen, co-designing, experimentation, and evaluation require back-and-forth interactions between scientists and users. But this cycle does not necessarily lead to a definitive innovation, because many new questions may arise at each stage of cycle. So, we have to start again with these new questions, test them, and evaluate them again within the same cycle framework. For example, in this study on vermicompost, new questions arise about the availability of some specific plant biomass, the quality of vermicompost, and its insecticidal capacity. So it's clear that participatory action research is an iterative process that takes place in cycles, enabling constant improvements. The advantages of such approach, including continuous improvement, are to reduce adoption time, risk mitigation, and cost reduction. Research action based on this iterative process appears fundamental to improving soil management practices in agriculture.